Good evening, everyone. I am Partha from the Biomix team. Would like to welcome you all for today's session. The speaker of today's webinar is Dr. B.S. Arasu, who is working as a research scientist in the Walter Reed Harvey Institute of Research at USA, and he will be delivering us talk on insights into protein export in Plasmodium. On behalf of the Biomix team, I would like to welcome our eminent resource person, Dr. B.S. Arasu. Our speaker is an eminent scholar in the area of structural and infection biology. Henceforth, I believe that he will be nourishing you all through his talk in this webinar. At this juncture, I would like to thank Professor Dr. B.S. Arasu for accepting my invitation and taking the time out of his busy schedule to join with us to this webinar. It's a highly honor and privilege for our Biomix team in hosting this webinar with a eminent resource person. And I also appreciate and welcome all the participants of today's webinar who joined with us. It's my pleasure to introduce our speaker, Dr. B.S. Arasu. Dr. Arasu Balasubramaniyam is currently working as a research scientist at Walter Reed Harvey Institute of Research, Washington, D.C., USA. He is currently working on pursuing structure-function studies of proteins export in malarial parasites during infections. He obtained his postdoctorate at the National Institute of Allergy and Infectious Diseases, NIH, National Institute of Health, Maryland, USA. He obtained his PhD in structural biology from Max Delberg Center for Molecular Medicine, Berlin, Germany. And also he worked as Junior Research Fellow at National Center for Biological Sciences in Bangalore. He did his Masters of Science in Biotechnology at the University of Calicut. His research interests falls on infection biology and structural biology as well. He was awarded with DBT JRF and CSIR JRF for his higher studies. With this brief intro, I would like to welcome our guest speaker, Dr. B.S. Arasu, to deliver a talk on insights into protein export in Plasmodium. Before this, kind attention to the participants. Dear participants, you have question and answer session at the end of this presentation. You are supposed to place your questions in the chat box. Myself here to put your queries to a guest speaker during question and answer session. Now I hand over the session to our speaker, Dr. B.S. Arasu. Sir, please.
Hi, Partha. Thank you very much for the kind introduction, and thanks to the Biomix team for putting this together. And it's been a pleasure to connect with Partha through LinkedIn, and then on to share my ideas and research, and also my thoughts about what I'm going to talk about today. And welcome everyone who is watching this webinar today. And I hope I can create some inquisitiveness in your mind about how can you fight or help fight malaria in worldwide. So again, so my goal of my presentation today is to create that kind of curiosity in you about what is currently going on in the field of malaria and how dangerous it is, how grave the situation has become over the past decade or so. So my talk is mostly going to be creating curiosity let's dive into my presentation.
Yeah, he joined, I think so. Hello.
Yeah, I'm here.
You are here. Vishal bro. Sir, you can start now.
Okay, great. So today I'm going to talk about, as I was mentioning before, about malaria and how bad the situation is, and what can be done as young researchers in the community. So I'm going to talk about one of the aspects of which Plasmodium, which is the malaria-causing organism, which is doing it in order to survive, which is the protein export. So I'll give you some ideas about how Plasmodium is able to survive in the human's cells by doing protein export. So first of all, as from India, most of us are from India, so we all know malaria is a disease. It's a deadly disease, but very few of us know that how dangerous or how infectious this disease is. In 2018, malaria killed almost 4,05,000 people, of which 2,72,000 were under the age of 5. And this is not just 2018. If you see the table below, it is it is through the last decade, it's been killing about 500,000 people. And looking at these numbers, I was very surprised when I looked at this number about like 5-6 years ago, and comparing it to the current our situation of COVID-19 deaths. I think we are nearing almost 1 million deaths. So looking at that, I was curious why malaria has not been looked after so well in order to contain or prevent this disease. One of the aspects which comes to my mind is maybe malaria doesn't have a conventional market to provide the therapy or provide the medicine. So that's one of the reason I believe that malaria needs more attention in terms of therapy and prevention. As you see here on the table, countries from Africa, they are the most affected countries, and this is one of the reason why I want to contribute in developing therapy or cure or prevention strategies for malaria. So some of you may not know like what causes malaria, like what are the species or what is it all about. So malaria as a Wikipedia definition, if you see, it's caused by an organism called Plasmodium. It's an unicellular organism, it's an eukaryote, and it's an obligate parasite. Parasite is something which requires a host. There are many species of Plasmodium which is known to cause malaria, but we are going to talk about the organism which is going to cause malaria in humans. So there are 4 mainly: Plasmodium falciparum, Plasmodium malariae, ovale, and vivax. Among the 4, falciparum is the most deadliest one because it has been killing so many thousands or hundreds of thousands of people in the past decade or even more. So Plasmodium. So Plasmodium, as I told you, it needs a host. So there are 2 hosts in the life cycle of Plasmodia. So one is the mosquito, and another one is us. So if you see the figure on your right, so this is a figure from the CDC. See here, it can be divided into 2 major parts. One is the mosquito stage, where the the parasite is living in the mosquito, and then the human stage. Human is, there are two organs or two systems it infects. So there's one is liver, and then it goes to your red blood cells. So to mention quickly how this life cycle happens is that when a mosquito, which is, let's say, already infected with Plasmodium, when it bites you, or when it bites you to have its meal, it sheds some of its infective stage parasite, and then it goes to your liver cells, and then it stays there for a while, and it multiplies, and then it ruptures, it lyses the cell, and then it comes to your bloodstream, and then it finds its way inside the red blood cells. So today I'm going to talk only about the red blood cell stage, or the erythrocytic stage of its life cycle. Why this is important? Because it's the stage where you can have all the symptoms associated with malaria: febrile, chills, fever. These are the less severe symptoms of malaria, but there are cases of severe malaria where the parasite can enter your brain and then can be very fatal. And this is one of the aspects of studying Plasmodium life cycle. So this is the RBC stage I'm talking about. So as I said, once it enters your red blood cells from the liver, there are many stages which the parasite has to undergo, meaning it has to develop. There are mainly three stages. One is called the ring stage, and the trophocyte, and then the schizones. So this is all happening inside the red blood cells. So as you know, it's an intracellular pathogen, meaning it is hiding inside the red blood cells, covering itself from the immune system of your human body. So Plasmodium has been studied more than a century, and then we know its life cycle, how it develops, or different stages of its infection. As you see on the figure on the right, the arrow shows, the red arrow which is facing down, lists the stages it has to go, the different morphologically different stages it has to undergo to become and to fulfill its life cycle.
Sir, excuse me.
Yeah.
You are not presenting, sir. Cannot see your presentation.
Oh my goodness. I'm sharing my screen, right?
No, it is not. Just press the start present now option.
Okay, wait, hold on. Did it got cut recently, or was it like this all the time?
No, it was from the beginning.
The beginning, or just continue?
Yeah, you can do it from the beginning, sir.
Okay.
Yeah, thank you, sir. Thank you. You can. There is an option, hide option, right? Yeah, it's okay. It's now all right. Yeah, That's I.
Okay.
Slideshow.
Full screen. Yeah. Can you see mine now?
Okay, sir. It's all right now.
Okay, man. Okay, let's do this. Okay, great. So today we are going to talk about protein export in Plasmodium. How Plasmodium, or the malaria-causing organism, is able to survive in the human, or how is it able to kill so many people by exporting its protein. One of the motivation I have to work on malaria is the table below. So in 2018, malaria killed about 4 lakh people, of which 2,72,000 people were, or children were under age 5. And this is not just 2018. And if you see the table below, it's been killing about 500,000 people for the last 10 years. And this is it's very grave situation. And you see most of the cases, or the people who died, are from African nations. It's about 380,000 people in 2018. And so there are many species of Plasmodium which is killing the humans. So what is the organism which causes malaria is Plasmodium, and then it's an unicellular organism. It's an eukaryote, and then it's an obligate parasite, which is it needs a host to survive. So there are 4 species of Plasmodium which is known to cause malaria, which is falciparum, malaria, ovale, and vivax. So as I said, like, it needs a host to survive. So there are 2 hosts. One is mosquito and a human. And you see the figure on your right. It can be divided into 2 major parts. One is the mosquito stage, and the other is the human stage. So today I'm going to talk about only the human stage, of which I'm going to only talk about the red blood cell stage, or erythrocytic stage. So to give you an overview, when a mosquito feeds on you to have its meal, it infects you with the infective stage parasite, which enters into your liver, and then it develops there, and then it ruptures the liver cells, and it enters your bloodstream. Once it's entering the bloodstream, it has to survive. It has to multiply. It has to make sure that it's evading the human immune system for it to survive, and then to continue its life cycle. As you see here, there are different stages of red blood cell stage malaria. And as you see on the right, there are pictures of human red blood cells which carries the parasite. So in order for the parasite to develop, it has to undergo different stages. The main stages are ring, trophozoite, and schizons. Once it undergoes its life cycle, it becomes it continues to infect more red blood cells, or it becomes gametocytes, which is picked up by the mosquito, and then the life cycle continues. So I would like to introduce or make you feel that why is it important to study protein export, and how complicated it is when an Plasmodium infects the human erythrocyte. On the picture, if you see, it's a schematic of an PF infected erythrocyte. The red shape is the human erythrocyte, and then the blue is the parasite, the Plasmodium parasite. So what happens when a parasite infects erythrocyte is that when it enters the cell, it covers itself with a it's a organelle called parasitophorus vacuole, or simply called vacuole, which is yellow color in this figure. As you see here, there are three membranes of this PF infected erythrocyte. One is the PPM, which is the plasma membrane of the parasite, and the PVM, which is the parasitophorus vacuolar membrane. It's again a barrier, and then finally the plasma membrane, or the membrane of the human erythrocyte itself. So as you see here, there are three barriers, and then this is how it fortifies its presence inside the human red blood cells. So why is malaria dangerous, and why it has to be studied? So that was one of the first questions which most of in the community are looking forward to address. So what is the pathogenesis of malaria? So when it enters the cells, it multiplies and then it ruptures red blood cells, thereby leading to anemic conditions. And also it has the capacity to adhere to your blood vessels, thereby blocking or restricting your blood flow. And then the third thing which it needs to do is to uptake nutrients from the red blood cells. Human red blood cells, we know it is it's a very good resource of hemoglobin, which is used by this parasite for its survival. And the most important, or according to me, it's one of the most important aspect of the Plasmodium parasite is to export its protein for its survival. Protein export is, what I mean is, it has its own genome, it has to produce its own protein, and then it has to export it out of its cell onto the human red blood cells for its survival. As you see here in the next picture, protein export in Plasmodium falciparum happens broadly in 4 or 5 steps. So first step is that the protein which is synthesized from the parasite has to cross the PPM, the Plasmodium or parasite plasma membrane, and then it has to enter the vacuole, the yellow space, and then it has to cross the PVM, and then it is in the red blood cell cytosol now. And then from there, it has to transport to its cell surface, or the red blood cell erythrocyte stage surface. There are few terminologies or the proteins which are well known to cause the pathogenesis of the malaria disease. One is PF, ENP1, and the knob structures. These are the physically observable structures on the infected cells. So one of the aspects I would like to stress upon, or even talk about entirely, is the PTEX. So what is PTEX? It's a translocon. It helps translocate protein across the membrane barrier, which is the PVM, parasitophorus vacuole membrane. It is called Plasmodium translocon of exported protein, hence the name PTEX. So why should we study PTEX? First question is PTEX druggable? That is, can we find a drug? Can we explore in aspects of finding a molecule or a small molecule in order to stop the function of PTEX, so that the life cycle of Plasmodia can be interrupted or inhibited, so that so many lives could be saved? That's the reason why we should study PTEX or its function. So as I said, it's a complex. It's a membrane protein complex on the PVM. To study it in more detail, we need to know what this complex is. So there have been reports for the past 10 years about the composition of this complex, and it's well-known fact that it has 5 different proteins. These 5 different proteins assemble together in different proportions on the PVM for the parasite to survive. So EXP2 on your right, it's one of the protein, PTEX 150, PTEX 88, HSP 101, and TRX2. These are the proteins which constitutes the PTEX complex, of which EXP2, PTEX 150, and HSP 101, they are the core components, the one in orange, purple, and green. They are the core components of this complex, and people have been trying to study this complex so that it can be exploited as a target for curing or prevention of malaria. So not long ago, like 19 last year, there been a cryo-EM structure of PTEX. Cryo-EM structure, most of you may be familiar or even heard of this technique. It helps to understand the structural organization and with great details. Sometimes it can go up to atomic resolution. The report in 2019 says that PTEX is a membrane protein which has confirmed the previous theories, and then it forms a megadalton complex. The whole molecular weight of this complex is about 1.6 megadalton, and the structure is known. If you see here on your left, I have marked as a PVM. This is the parasitophorus vacuolar membrane, where the EXP2 protein is green in color. It is inserted into the membrane, and then PTEX 150, which is sandwiched between HSP 101 and EXP2. So this is a well-known fact now that this complex helps in exporting protein. I'm saying this because they also observed cargo. I call it cargo because that's the product which has to be exported out of the parasite for its survival. So the cryo-EM structure of the PTEX complex observed cargo in transit. So this complex has a pore in the middle so that it can help transport or translocate protein through this pore. On your right, you can see there are 4 images. These are the classes, 2D classifications. I'll come to this later in my presentation. So this is how the complex looks under electron microscope. As you see here on the bottom left, it forms like a potato shaped or a spindle shaped structure. So it can be easily incorporated or embedded into the PVM. And then it can orient itself and help export the protein into the cytosol of human red blood cells. So the first challenge is how should we study this complex? Because it's inside the parasite, and it won't be. There are a few challenges associated with studying proteins from a parasite with respect to folding and its function. So one of the goal is to study the native protein complex, which is the PTEX complex present in the parasite in the human red blood cells. So one of the ways is to purify the PTEX complex from the parasite itself. And this is a scheme of how a purification strategy works. So as you know, PTEX is it's a membrane embedded complex. So in order to keep it happy or keep it functional, we need to isolate or pluck it out of the membrane in its native state. And one of the strategy is to use detergents. If you see in the picture on my left, there are micelles, which has blue in color. So, and the membrane. So they are analogous. I will tell about the properties of lipid bilayers in the coming few slides. So what you have to do is you need to isolate or remove the PTEX from its lipid bilayer conformation in a solution with detergents. So that is called solubilization, wherein you provide a right amount of detergent and the right detergent for it to keep its functional state. And then once you have that detergent and the right amount of detergent, you can test for its functionality and correct conformation stage. So there are many ways. So one of the ways I did to purify the PTEX components is to the use CRISPR. CRISPR is a way of editing your genome. So using the help of my colleagues, I created a parasite or Plasmodium falciparum parasite, which has different tags, affinity tags for helping the purification. One is his tag, flag tag, thrombin tag, and so on and so forth. So using this tag, one can purify the endogenous protein complex from the parasite. At the bottom left, you can see these are the images of a native gel. Native gel, most of you know, it's a gel to estimate or give an idea about the molecular weight of the entire protein complex under native condition, that is without SDS or under non-denaturing conditions. As you see here, I've tried like about 20 or 19 different detergents and different concentrations. And after lots of optimization, I found a detergent which is optimal for keeping the function and also the structural integrity of the complex. So once you have your protein complex, you need to run a SDS page to see what are the proteins present in your tube, or what you purified from the parasite. And when you run it on a gel, I identified PTEX 150. I'm talking about the gel on your right, PTEX 150, HSP 101, and EXP2. So we confirmed this protein by mass spectrometry that this is indeed the components of PTEX. And now we want to proceed how they look under native condition. So one of the initial stage or the first stages of getting the structure of any big protein complex is to do negative staining. Negative staining, very quickly about what happens in negative staining is that you need an electron microscope for sure. There are tiny copper grids where you apply your protein and then you stain the grid with uranyl acetate. That's one of the stain which I used to stain the protein complexes. So it's called negative stain because the protein does stain the background and then leave the protein particles standing out. So it gives you the protein complexes gives you a white appearance in the electron microscope. So as you see here on the picture on the left, this is a electron microscopic view of a protein complex, and you see here there are a few white dots and white circles or white spindles you can observe if you pay a closer attention to it. So there is a red box which is a inset of a zoom in of what I was seeing in the microscope, and you see in this dotted rectangles. You see a donut-shaped structure and also like a potato-shaped structure. So these are the characteristic feature of high molecular weight protein complex. So as I said, what are these 2D classes, or how the electron microscope or the software package which comes with it is able to isolate or identify the particles of the protein. As you see here, once you have this picture from my previous slide, there are thousands of particles, sometimes even more. So it's not possible for a human eye to pick or locate these particles individually. So what the software does is it has algorithms to pick shapes or dimension of a particle, and then it puts them together. Let's say out of 100, let's say there are 100 particles, and let's say out of 100, there were like 20 of them are similar in shape. So it makes a classification of that shapes, and then it gives you a two-dimensional classes or the 2D classes of your protein complex. As you see here on the picture, on the panel on the left, there are about 100, I think more than 100 2D classes. And of those 100 on your right there are three major classes, which is very suggestive of what PTEX might look in solution. On the top square, you see here there are it's like a flower-like pattern, and you see like 7 globular parts. It's a very suggestive of EXP2 being that in that complex. And then the next 2D classes, next two 2D classes are most likely the entire complex which was observed in the negative staining of EM. So we have the structure, we have the complex purified. So how can we exploit it to, as I said, like how can we exploit it as a target for therapy for malaria? There are 2 ways. Actually, in fact, there are many ways. So one of the 2 major ways are, one is cell biological way of addressing the function of this complex, and one is the electrophysiology. Electrophysiology is, as you might be knowing, it's a study where you characterize its physiological properties, like how is it responding to applied current and applied voltage, and what are the ions it is conducting, or what are the solutes in terms of survival it's helping transport on a membranous structure. So I would like to address this characterization of PTEX through electrophysiology. So how does this electrophysiology work? If you remember my previous slides, so first we need to isolate PTEX or purify PTEX from the endogenous membrane. You need to solubilize. You need to purify out of the membrane. Now we need to study its function. So we need it to be incorporated back into a lipid bilayer, and this is called reconstitution, where you shed the detergents and then put it back in an artificial lipid bilayer. So moving on, what is a lipid bilayer? It's a textbook definition. It has an hydrophilic head and hydrophobic tail, and it forms 2 layers, the tails facing each other and the polar hydrophilic heads facing on the opposite side. Thereby it creates a barrier. Now, so there are many lipids or phospholipids found in human cells, but one of the lipid or a phospholipid I used to create an artificial membrane is this one shown at the bottom, DPHPC. So it's an organic, it's dissolved. This phospholipid is dissolved in solvents like decane or octane, and then you can artificially create these bilayers in a given setup. So to study this, I used a technology which is available in the market, which is called Orbit Mini. So it's a small device. It's about the size of, let's say, a computer mouse. And then it's plugged into a laptop, and then you can work on it to study the function of your protein complex. As you see here, it's about the size of a coffee mug, and it has a few parts. And I'll tell you, I'm not going to go into the technical details of this setup, but I would like to explain the principle how this technology works to study protein function or electrophysiology of this protein function. So there is a chip called Mica, microelectrode cavity array chip, and it's about the size of a coin. It's just green in color, and then it has a cavity in the middle. And then if you look closer into this cavity, there are four cavities, subcavities, I would say. And then the diameter of this cavity is about 50 to 150 micrometers. It comes in different dimensions. So on the picture on your left, if you see, the blue wall is the cavity, and then the one which says lipid bilayer on the picture is what you artificially create, where with using the DPHPC as a phospholipid. And then we call it painting the lipid bilayer. You take your phospholipid in a brush or a Teflon wand, you paint on the surface of this cavity, and then you create a bilayer. And then once you have your bilayer, it's time for you to add your protein of interest, in this case PTEX, and then wait. What happens now is the protein diffuses into this cavity, and then sheds its detergent, and then incorporates into this artificially created lipid bilayer. And if it's functional, or if it's able to conduct ions. So another aspect which I want to point out here is that so we have created a barrier using this artificial bilayer, one which is below the bilayer, and another one above the lipid bilayer. So the components or the contents of this lipid bilayer across this lipid bilayer can be controlled. So usually above the lipid bilayer is your buffer, which has your buffer and different sodium chloride concentration and other aspects of studying the protein complex. So once you create your lipid bilayer, you have your protein ready, and then it's time for you to add it to the setup, and then wait for it to incorporate. Meaning it has to diffuse, and then it has to adjust itself in such a way that it can bring back its functionality from what it was in the parasite. So I'm going to simplify what one records of an electrophysiological signal from a channel. This graph is very simple to understand. On the left, you see it's an axis diagram. On the left, the y-axis is the current in picoamperes, and the x-axis is the time in milliseconds. And you see here this black trace which runs on the x-axis. It has stages which goes up, then remains in the up stage, and then comes back, and then it goes on. So as a convention, there are ways to address this activity or how this channel is functioning. So on this device, you can apply potential, like a positive potential or the negative potential. So the convention is when you apply, let's say, 10 millivolts, and when the channel is functional, it opens. And then there is you can observe the current passing through the channel. And this green dotted line, which is on the top, which says that the channel is open, meaning there is some transport of ions. And when it comes down to the red level, you see that the channel is closed because there is no current flowing on. So it's the opposite when you apply the negative potential. So the upper level is closed, and then the level at the bottom is open. So this is one of the convention we follow to study the characteristic properties of the channels. So once you have your protein purified, your device is ready. When you put your protein, what you cannot control is how many channels, how many protein molecules is getting incorporated into the lipid bilayer. There could be 10, there could be hundreds, or there could be even one. One of the way to address that problem is to look at your traces from your recordings. As you see from the recording on the top, there are two dotted green lines suggesting that there are two open levels and then there is one closed level. So this is very suggestive or a characteristic feature of two channels present on the lipid bilayer. But what we are interested in is that we need only one single channel which we can exploit or understand how this channel is functioning in terms of applied potential. As you see at the figure down, there are only one levels, open and closed levels, and then the channel is responding to your applied potential. So it's a kind of a signature of this molecule. Wherever you record this channel, it should have this characteristic feature. And this is the property which people would like to, or I would like to address. Once you have the signature of this molecule, how can we exploit it? How can we help stop or inhibit its function using different ways of chemistry or drug discovery or any kind of therapeutic intervention? So, as I said, so there are many ways to address. So one of the ways to address or address these aspects of druggability of PTAX is to ask what are the ions or what are the solutes is conducting through the channel. Let's say whether it's conducting chlorides, whether is it conducting sodium, or is it conducting amino acids or other solute molecules. And another aspect is called gating analysis. So as I told you, like the channel is open and closed. One of the aspect of channel is that how long the channel was open, how long or what is the duration it has been closed. So these are the characteristic feature of any channel. Once we have these information, like what are the ions it's conducting and how long it's been functioning, how long it takes to open, how long it remains closed. So once we have this information, we can exploit it in terms of identifying molecules which can inhibit this function, and thereby we can use this as a candidate or a potential candidate for treating malaria. So I'm almost coming to an end of my presentation. So having studied PTAX, and I think there is still scope for developing new technology. So one of the aspects I could think is why couldn't we couple electrophysiology to super resolution microscopy? So once we have this technology, what I envisage is that we could exploit the properties of electrophysiology, as I mentioned, like what are the ions it is conducting, how long the channel is open, and then to a super resolution microscopy where you can isolate or visualize protein complex in a nanometer resolution, where you can study the channel in a single molecule state. So once you identify this channel using, if there are any.
Yeah, thank you so much, sir. Your presentation is quite interesting and so informative. It proves that
Thank you. Yeah.
You fascinates the field of structural infection biology. You correlated the structure with infection aspects, right?
Right, correct.
Yeah. So today we had a mass number of participants throughout this webinar, and a few of the questions are, I am reading out the questions from our participants.
Sure.
The first question is from Sivapriya, that she is asking, can you elaborate how CRISPR techniques helped your work in your study?
Okay, sure. So CRISPR, it's a genome editing tool. So every organism has a genome, and then it's a nucleotide sequence. So what we would like to address this aspect of research using CRISPR is we wanted to tag or insert an amino acid sequence onto this protein, be it any protein. It's not applicable only to PTEX, but it's also applicable to any protein. Let's talk in terms of genome, nucleotide sequence. We can edit this nucleotide sequence of this organism endogenously. So it's CRISPR is a well-known or well-established technique now, where you have your plasmid, which carries your gene of interest or sequence of your interest, and it has some signature motifs, and then it has to be transfected into your cells, and then this plasmid goes into your organism, and then it cuts. I'm going to give you like broad idea for how it happens. This is called PAM sequence, and then it has to be addressed right onto that spot where you want to edit your genome, and thereby you can exploit it. So what I did in my research is that since I wanted to address this complex, I wanted to put an affinity tag. As I showed, it's a his tag or a flag tag. These are the nucleotide sequence which will help us purify the complex when they are translated. So I exploited the flag affinity technique. So using the CRISPR technique, I had the nucleotide sequence of his tag. I had it in my plasmid. I transfected my parasite containing cells, and then it takes a long time to give you an idea to get your parasite which has a his tag. It takes about any time between 2 to 3 months to get your parasite clone, we call it. Yeah, and that's one of the approach where you can exploit CRISPR to make parasites of your interest.
Yeah. The next question is from Abraham, that he asked, you have mentioned that about the detergent in your work. What kind of detergents did you use for purification, and what are its compositions?
Okay, nice question. So as I showed you, like there are many detergents, like there are hundreds of detergents out in the market, but based on the characteristic, I narrowed it down to like 20. One of the main detergents I characterized was Triton X100 and GDN, These are the different characteristic detergents which I used, and one of these two detergent has always worked for me in terms of its keeping the function of this complex. And it's a lengthy process. You need to start based on the CMC, this critical micellar concentration of any detergent, and then you work your way through in terms of its concentration. To give you an idea, let's say Triton works anywhere between 0.1 to 0.7%. 1% is sometimes working, but you need to, find out. It's very subjective. Each protein behaves differently, and Triton, for example, in my hand, it's one of the best behaving detergent. It also kept the protein functional, and it also held the complex together. The cryo-EM structure, which was published of the PTEC structure, was solved using Triton X100. Partha.
Yeah. The next question is that, so there are many ion channels. How do you identify the particular specific ion channel is used for exporting and importing parasites, toxins inside and outside? How do you identify a specific channel?
Great, good question. So how this all started is this was in 2009 from a group in Australia. So until then, people had only thought about the survival aspect or the protein export aspect of this Plasmodium. So the first, they set a few criteria, like what could a protein complex should have in order to export protein or pump the protein out of the cell. The first aspect is that it needs to have energy source, meaning it has to actively export the protein across the barrier. And then the next aspect is it should have like a protein which should have ability to form a pore or a channel on a membrane. And then they also envisaged to have adapter proteins. So these two proteins has to be supplemented by other proteins. So this was a 2009 report. They first looked for proteins or protein complexes which had these two aspects. One is the energy source, and then the one which has the property of making pores. So when they looked at the genome, and then they know that this has to be on the PVM, because the exporters on the parasite membrane is well known, and the only aspect of finding a protein source is on the PVM. So they isolated, they tagged the different proteins. They knew that EXP2 could be one of the pore formers, or I think I would say they just guessed it. And then the next aspect is the HSP101. It's a heat shock protein. It's a typical chaperone which helps protein fold and unfold. It has it's an ATP binding protein. ATP binding is very characteristic feature of an protein which exploits the energy of ATP hydrolysis. So when they identified these two aspects in their let's say pull downs, that's what they did. They tagged the HSP101, and then they saw what are the other complexes or other proteins it's taking it apart, taking it together. So once they had this, they characterized this whole complex. HSP101, it had EXP2, and then it had the PTEX 150. So when they identified this, they didn't get it right. What I mean by that is that they didn't know how many HSPs or how many EXP2 is in this molecule until recently in 2019. We exactly know what these proteins are, how many proteins it is composed of. We at least we know the stoichiometry of this complex. Yeah, I hope I kind of addressed your question, Partha.
Yeah. It is reported in 2009, right? Do you know in which article it has been published in 2009?
It's a paper from Tania D. Koning for in 2009 Nature.
Okay, so fine. It has been published in Nature, right?
Yeah, 2009. That was the first report of an or an idea of there could be an export complex. It's an Australian group.
Yeah. Since you are from structural biology, have you applied any biophysical work techniques, biophysical techniques in your work, like NMR, all these things?
No, I didn't apply NMR. One of the aspects, biophysical techniques are broad. It's you can list a few in which comes under this category. One is I would say biolayer interferometry. So this is something which I applied not to study this protein complex, but it can be studied. So one of the aspects of this complex is, as you know, it has like 5 different proteins, but we don't know the affinities. For example, how tightly these proteins are binding to each other. So one can exploit or one can use this biolayer interferometry. So this is a method which can be used to address this kinetics, like how what is the on-off rates of these two proteins or proteins in any different combination. And this is one aspect you can address this. And then another aspect is DLS, differential light scattering. This can be used to address the size or the distribution of your size of your particles in terms of nanometers. So you have your solution. You have your solution in your tube, and then it has your protein of interest. And then when you put your sample in this device, it's differential light scattering. I give you a broad idea of how this functions. It calculates the or estimates the particle size based on its Brownian motion in the solution, and it gives you what is the size distribution. Let's say 20% of your sample is of particles which has let's say 500 nanometer in diameter. So this is one aspect you can study. And I think these two techniques can be very helpful. And cryo-EM is one of the hardest field of finding out the structure of protein complexes in atomic details. Yeah, Partha.
Yeah. Yes. Thank you, sir. What are these thermodynamic aspects of your proteins that you export? Have you studied any thermodynamics aspects?
Not really. No, I didn't study thermodynamics. So, but what you can do is, as I said, like you can use biolayer interferometry, or you can use ITC, isothermal titration calorimetry, to get your thermodynamic characteristic features of your protein complexes.
Yeah. Thank you, sir. Thank you so much, sir, for your time and patience in listening and answering our questions. Thank you for spending a valuable time with us. My sincere
pleasure.
My sincere gratitude to you on behalf of my entire team. Thank you so much, sir.
Thank you, Partha, and thank you everyone for listening, and thanks to the team, and thank you very much, everyone.
Yes, I hope all participants are satisfied with the speaker's elaborate explanation in answering all your questions. Thank you so much. I thank everyone who joined with us through this webinar. A few announcements to the participants. The participants, you can get your certificates from the website and you can go to the information page of a website, Biomix website, so that you can download the certificate with after 3 hours of the completion of this webinar. Thank you so much. Stay connected with the Biomix team for further updates. Join with us. Stay tuned. Thank you. Thank you, everyone.